Welcome to our exploration of animal diversity. Today, we are going to uncover the fascinating and somewhat terrifying life cycle of Taenia solium, commonly known as the pork tapeworm. Imagine a microscopic stowaway that travels across the world, not by walking or flying, but by hiding inside one vehicle, waiting to be transferred to another. This is a perfect example of parasitic adaptation, and understanding it is crucial for protecting human health. Let us dive into this incredible biological relay race. Now that we know what our topic is, let us understand why this parasite is such a master hitchhiker. In the animal kingdom, survival is everything. The pork tapeworm has evolved a highly specialized strategy where it absolutely cannot complete its life journey in just one environment. It requires two completely different living hosts to survive and multiply. By exploiting the eating habits and waste disposal systems of other animals, it ensures its continuation. Understanding this complex relay race is the first step in learning exactly how to break the chain of infection. Building on this idea of a relay race, biologists formally describe this life cycle as digenetic. Digenetic simply means that the parasite requires two distinct hosts to complete a full generation, from an egg to an adult. For Taenia solium, the primary, or definitive host, is the human. This is where the adult tapeworm lives in the small intestine, absorbing nutrients and reproducing. The secondary, or intermediate host, is the pig. The pig ingests the eggs from the environment and harbors the larval stage in its muscle tissues. Before we trace the exact steps of the cycle, let us look at the physical structure of this parasite. The adult worm is perfectly adapted for its environment. It features a head-like structure called a scolex, which is equipped with hooks and suckers. These allow the worm to anchor itself firmly to the human intestinal wall. Following the scolex is a long chain of reproductive segments known as proglottids. As these segments mature, they become essentially egg-making factories. Eventually, they break off and leave the human body through feces, ready to start the cycle all over again. So, how exactly does the parasite move from host to host? The biological cycle begins when an infected human passes feces containing mature, egg-filled segments called gravid proglottids into the environment. If pigs forage in these contaminated areas, they accidentally swallow these microscopic eggs. Once inside the acidic environment of the pig's stomach, the tough outer shell of the egg dissolves. This releases a tiny six-hooked embryo known as an oncosphere. Using its hooks, the oncosphere bores through the pig's intestinal wall, enters the bloodstream, and travels directly to the pig's skeletal muscles. Following the embryo's journey into the pig's muscles, it settles down and undergoes a transformation. It turns into a fluid-filled sac called a cysticercus, or bladder worm. Pork containing these cysts is commonly referred to as measly pork. The cycle is finally completed when another human eats this infected pork, raw or undercooked. Once ingested, the cysticercus wakes up in the human intestine, attaches itself to the gut wall using its scolex, and begins growing into a brand new adult tapeworm, ready to produce more eggs. While the standard cycle involves pigs, there is a dangerous twist that makes studying this parasite critical for global public health. If a human accidentally ingests the microscopic eggs directly, perhaps from contaminated water or unwashed hands, the human acts as the intermediate host instead of the pig. The embryos hatch and travel to human tissues, including the brain. This causes a severe and potentially fatal disease called neurocysticercosis, which is a leading cause of adult-onset epilepsy in developing nations. This is why mastering this life cycle is key to preventing neurological diseases. To understand why this complex and risky life cycle is actually successful, we must look at the sheer volume of eggs produced. Because the chances of a pig eating a specific piece of contaminated grass are very low, the tapeworm compensates by releasing millions of eggs. We can model this using a simple multiplication formula. E total represents the total number of eggs released. NP stands for the number of gravid proglottids shed per day. EP is the number of eggs per proglottid, and T is the time in days. If either the segments shed or the eggs per segment increases, the total environmental contamination skyrockets. Let us put this formula into practice by calculating the reproductive potential of a single adult tapeworm over 1 year. We are given that the tapeworm sheds an average of 5 proglottids a day, and each contains 40,000 eggs. We want to find the total eggs released over 365 days. First, we multiply the 5 proglottids by the 40,000 eggs to find the daily output, which is a staggering 200,000 eggs every single day. Next, we multiply this daily figure by 365 days. The final answer is 73 million eggs per year. This massive number explains why poor sanitation easily leads to widespread infection. Now, let us look at the mathematics of transmission from the pig back to the human. A villager will only get infected if the pig they are eating is infected, and they fail to cook the meat thoroughly enough to kill the parasite. Because these are two independent events happening together, we multiply their individual probabilities. P infect is the overall risk of contracting the parasite. P pig is the chance the pig is infected, and P cook is the chance the meat is undercooked. This mathematical approach helps epidemiologists understand where the weakest links in the transmission chain are located. Let us apply this probability rule to a specific village scenario. Suppose the probability that a slaughtered pig is infected is 0.15, or 15%. The probability that a villager undercooks their pork is 0.20, or 20%. We substitute these values into our model, multiplying 0.15 by 0.20. The final answer is 0.03, which translates to a 3% chance of infection per pork meal. While 3% might seem small, if a person eats pork frequently, the cumulative risk becomes dangerously high over time. Having calculated the risks and reproductive rates, let us visualize the entire process. Here you see the complete biological relay race. At the top, the adult tapeworm thrives in the human intestine. Follow the arrow down, and you see the eggs passed in human feces. Next, the pig ingests this contaminated food. Inside the pig, the eggs hatch, migrate to the muscle, and form cysts, creating measly pork. Finally, the cycle loops back to the start when a human consumes this undercooked pork. This visual perfectly summarizes the digenetic pathway we have been discussing. We have covered a lot of ground today, so let us review the most important ideas. First, Taenia solium is a digenetic parasite, relying on a strict relay race between humans and pigs to complete its life cycle. Second, it compensates for the high risks of this journey by producing millions of eggs, ensuring its survival through sheer numbers. Third, if humans accidentally ingest the eggs directly, it leads to the dangerous condition known as neurocysticercosis. Ultimately, understanding this cycle and the math behind it empowers us to break the chain of infection through better sanitation and proper cooking. Great job following along.